Okay, hi everyone. Welcome to our next webinar. So this is, I think, webinar 12 we are organizing as Youg ISA. So our next speaker, this month's speaker is Gabriel from Hamilton Institute, Maynooth University. So Gabriel, correct me if I'm wrong, but Gabriel has done a degree in biology from University of Sao Paulo, and now he's doing a PhD in data science at Maynooth University. So Gabriel is going to talk about a new machine learning methods called pattern-based prediction for predicting insect outbreaks based on a time series of aphids, pest species of cereal crops, and more. So Gabriel, the floor is yours, and we are all looking forward to hear your presentation. Thank you.
Thanks a million. Thanks for the introduction and for the opportunity to be here. Very happy to do that presentation. And again, many thanks for that. So during the presentation, I will be looking there because I have two screens. So if I'm looking there, it's because of that. Okay. And basically, that's the idea of the presentation today. I'm going to be talking about the pattern-based prediction and the machine learning methods for detecting population outbreaks. So without further ado, I'd just like to introduce you what I'd like to be covering here today. So first, I will talk in terms of the introduction. And in the introduction, I will be talking about insect outbreaks, what I'm talking about insect outbreaks, what they are, and some concepts about it. Then I talk which type of machine learning methods I will be using, type of learning more specifically, and then the objectives of the presentation. Then I go directly to detecting outbreaks based on pattern-based prediction, and I show you the methods, the results, and finally, I mention the conclusions. The references will be in the end as well. So just like to make an illustration of what I meant about insect outbreaks, and thanks to BBC, I could show that to you today. So this is a video that BBC was made about an outbreak of locusts. So basically, this is a phenomenon that's happened in some places here in the world, and we have basically a lot of individuals occurring at the same time. So as you can see in a couple of seconds, these massive individuals, they basically are going to migrate to certain locations, and in the middle of the path, they're consuming a lot of grass. And if some crops are in the middle of that, they're going to consume everything. There are also some videos, very interesting, if you like to have a look in YouTube, about what happened when those insects go to the cities. That could be a little bit annoying. You can see if you're walking and imagine an insect of this size going in your face, not a good idea as well. But here I'm going to be covering more about the economic impact of that phenomena. So as you might imagine, this amount of insects could cause a lot of economic damage if they go directly to a crop, for example, a wet crop, as you can see in the video. There's a variety of locations that we are, not necessarily wet crop, but we can see that they could cause a lot of damage. So ideally, if we could have a certain weeks or a certain amount of time before to advise the growers that, well, we can have a certain outbreak happening, would be much interesting and useful for them. So basically, that's the context of what I meant about insect outbreaks. However, we can have insect outbreaks in a different context as well. The previous one that I mentioned was a very strong event, extreme event with many insects happening. But in agronomy and agriculture, we can have different species as well, having different levels, number of individuals that we can still classify as outbreaks. So for example, if we had some other type of species, for example, Chrysodexis includens, that is a species that basically can occur in some areas in Brazil, in my country, that we have a lot of those insects causing a lot of damage to certain crops. But here we'll be covering more about wet culture. In wet culture, we have aphids, and the aphids basically, they can study the biology of those species and understand, well, what would be the number of individuals that can cause economic damage. So before I can show you here some of the references, like since 1959, the concept of economic threshold has been covered and introduced. And the idea is the amount of individuals that can cause damage for growers. So basically, it's very specific for a certain pest species and for certain crops. So basically, you compute the amount of insects that can cause the damage and what could be impacting in. So the decision-making of the growers, so this decision-making in terms of what do I do to reduce this population, is based on that threshold. So once you have these amount of individuals in the certain field, you can see it and you apply any type of technique in order to reduce. These techniques can be, for example, biological control, which you can use predators. We can use parasitoids that are little wasps that they lay their eggs inside of the pests, and these egg hatches, the pest die and a new wasp comes out of it. Or you can use chemical like pesticides and other, just to reduce the population. So in this context, we're going to be covering more about wet culture. But to come up with the method in order to predict when we're going to have that outbreak or not, we're going to use the concepts of machine learning. So this is, I'd like to just bring a brief idea of what of machine learning we're going to be using here. So again, in machine learning, we're going to apply some specific tasks and make a machine to predict or automatically solve a specific task. In this context, the task will be predicting an outbreak or two events, a binary problem. There is or there isn't an outbreak. In this context, we're going to be covering here, among the other types of learning that we have inside of machine learning area, we could be looking to two of them that basically are going to be covering the supervised and non-supervised learning. But in the method itself, we're going to focus on the supervised learning. But just to make a comparison between those both, I'm going to make these introduction based on that. So it's basically the idea, the problem that we're going to face is, again, we have a grower very upset about the fact that he has a lot of locusts in his crop, causing a lot of economic damage, and we can compute and obtain some variables. So monitoring the abundances of individuals and monitoring variables would be very important for those algorithms. And we can say that, well, in the moment that the outbreaks is happening, I collect those variables. And also, if the outbreak's not happening, I will still collect the same variables in order to feed a certain algorithm that basically will adjust its parameters. And luckily, based on a train set, we will have a general algorithm that when I see unseen data, I could still come up with a prediction if it is or it isn't an outbreak in this case. However, this was just for supervised learning. If you go to non-supervised learning, we don't have the labels presented to the method, to any algorithm that we're using, but basically coming up with some clusters and trying to only based on the data, try to split into possible clusters. But as I mentioned earlier, here we're going to cover and the base will be based on supervised learning. Well, given that said, we basically here we intend, in terms of the objectives of that presentation, I'd like to first introduce the new method that is capable of predicting insect outbreaks and successfully, and also we can illustrate it to be interpretable. So that's the main idea. And to do that, we will illustrate our methods with some results based on a time series of insects, basically cover aphids, as you can see in this image, aphids in the wet culture. So given that said, what is an aphid? Well, aphids, now thanks to Cuppert for that video again, I can show you what it is, because these insects are very tiny, but they can cause a lot of damage. As you can see, these aphids, there's lots of insects and different species included in this genre. But the idea of them is basically they will be fixed on the plant. And as you can see here, this is the feed distillage. Basically, they use this to assess the phloem of the plants and consume their amino acids. They're going to use that for their development. Their development is simple. As you can see in the next second, they're basically a nymph stage and they grow into adults. And normally they keep aggregated in this structure in a plant. Usually you will see these insects, they're very small, but they can cause a lot of damage because of two reasons. The first, the fact that they're feeding on the phloem of the plant, they can weaken the plant, and also the fact that it can transmit some diseases. They transmit the diseases, and this disease will be the main problem. So in our case, we're going to have some species and passes specifically that cause a lot of damage by this transmission of certain viruses that can infect those plants. But you might imagine what would be the problem. They're so tiny in staying on the leaf, for example, how they can infect and be a problem in terms of outbreak. So what happened is, as you can see, when they are here or aggregated, there is a part of their development is that they can have wings and fly to different hosts. When I mean hosts is the plant itself. And basically when there's an enemy, or if you apply any pesticide, they communicate with themselves through pheromones, and they basically message in themselves saying, hey, I think we should move. And then the next generation start born with wings, and those will be able to spread over the crop and keep causing more damage and spreading, in our case, spreading the disease. So this is the example that we're going to have. This is a time series that's being collected in Brazil. This is basically a time series of aphids. The idea of the experiment is we have a crop, we have 4 traps. Each one of the traps will have the bonuses of the individuals. So this monitoring system is basically each week I go and specialist taxonomists will identify the species, count them for each one of the traps, and we sum them up the number of each one of them. So the time series that we have is basically per week, we're going to have the sum of the number of individuals per trap. So each week I'm going to have the number of them. As an illustration to what the time series is and how is the shape of it, the patterns that we can encounter, I show you in the next slide more details of it. But just mentioning about that now, in one example of important past, if you're interested, is the Hopalasiphum paddy, that species is the one that's most important in terms of transmission of diseases. So, but in this problem, we're trying to look to the sum of all the species and see if we're able to identify an outbreak or not, because we still consider them all. So looking to this outbreak, this time series, we have the following idea. So as you can see, I have 2 dynamics. In green, I have the regional dynamics, and in black, I'll have the decomposed dynamics. This decomposition is based in the empirical mode decomposition, and we use this empirical mode decomposition as an alternative to try to generalize our method. So basically, we can use in the method the original time series or the decomposed time series. It's just an illustration. So here, as you can see, we have the aphids, the sum of the aphids per traps per week. And here we have the threshold. So in red, we have the threshold. This is 200. As I mentioned earlier, we come up with this number based on specialists of the area. They discuss, there are some experiments to understand which would be the best number to have expressive economic damage. So this is where the number came from that discussion. So we have this threshold, and ideally, we will have certain moments in the time series in which we have the phenomena of outbreak happening. So if we have bigger values than the threshold, we will classify it as an outbreak. If it's lower values than the threshold, we classify it's not an outbreak. So this is our binary problem in that context, given the application of this problem. So here, in terms of statistics and what we're going to compute in this problem, I would like to show you the confusion matrix. So basically, we're going to have 2 it's a binary problem. We have outbreak or not outbreak. We have the observed data and the predicted. What we hope is that we can compute the true positive and true negative, and also have the 2 possible errors, the false positive and also the false negative in this case. So using this confusion matrix, we can compute the following statistics. The first one would be the true positive rate. That is basically the first column of the matrix that I showed you. So we're going to look for this column here. This would be the true positive rate divided per the true positives plus the false negatives, and the false positive rate, that is the second, focusing on the second column of the confusion matrix. Also, we can have the accuracy. That's what I'm going to show you in the end. The result, the accuracy of the method. That's basically true positive plus true negative, the diagonal of the confusion matrix, divided per the all frequencies, the sum of all frequencies in the confusion matrix. Also, we can have the ROC curve. basically. So using the two statistics, the first two statistics that I mentioned, we can have the rock curve. So basically we compute them like that. We have the true positive rate in the y-axis and the false positive rate in the x-axis. And ideally, if we have a good method, it tends to have an area under the rock curve close to 1. So as close to 1, the better the algorithm is. So just a little recap what we have. So this is the time series, the threshold that we're looking at, outbreak above, not outbreak below of the threshold that was indicated by our collaborators in the area. So using this, what we do is the following. So given the moment when the outbreak occurs, so I reach the threshold, I will look, now the name is patterns, so I'm looking for patterns here. To find patterns, we can basically go n points before the event happened. So let's say in this part here, I go and reach these observations, so I can go back m observations. So basically this first event happened, go back m observations. It happened again, go back m observations. And I repeat this every time that I reach a certain event. That event would be an outbreak, equal or the observations, the number of individuals being equal or bigger than the threshold. When this happened, I can summarize this as a matrix. Basically, as you can see, we have the xt. So xt will be the abundances in the time t. So this one here is related to the first event. So when I mean event is when the outbreak happened. So this would be the first event. And then I'm looking for n observations before of the event. So basically I look into 1 to m observations before, and we have one pattern. I look to the second one when it happened at outbreak. I select the observations before, store it in another row, so on and so forth, up into the I pattern, the I event that happened. So this we call the pattern matrix. This is what come up with the P. And also based on that, we can do the following. So look, we're gonna have these patterns through each one of the rows representing, but we can cluster them. Sometimes we have some patterns that are more related to each other. So ideally, we like to cluster them to see more clearly the distinct patterns that we have within this matrix. So doing that, we do the following. So I'm going to call I and J two patterns, or we can see the role of that matrix. So if they are sufficiently similar, we're going to group them, basically. So to group them, we do the following. So we have this certain association metric that we call D. So the P, I, and Pj, basically two rows of the matrix that I just showed you, we compute by 1 divided per C, that is the Canberra distance of Pi and Pj plus 1. So the Canberra distance has this following idea. This is how we compute it. And this is good for counting data as well. And one thing I'd like to note, to mention to you, is that this Canberra distance, when the Canberra distance tends to infinity, we have that this association tends to 0. And if the Canberra distance tends to 0, that the points, the patterns are very close to each other, then the association goes to 1. So using this association, we start the method. So first, get the matrix, and then you will come up with the clusters. Just an illustration of the clusters, we will see the following. So here you're going to have each one. So I selected m equal to 5. Let's look in 5 observations before the event. And you can see each row is being plotted in the graph here. So will be the matrix B. And then we can classify them, create clusters based on this, as I mentioned earlier, using the association. And then we come up with 3 other matrices. I call in these matrices P prime C. Okay, so C will be the number of clusters that we found based on the value of the cluster to choose. So this case, I chosen for an example. So you might imagine, what is this observation? So these observations comes from the Ricker map. So basically, I simulate values from the Ricker map that it was used from the original literature in 2011. And I use R, that's the growth rate, equal to 4. And I use K, that is the carrying capacity of the model, equal to 2000. And I simulated 1,000 observations from that. So this is why you can see those. So the 2000 here is related to the carrying capacity from the Ricker module. So each one of them, we can have the classes. the clusters in here. So each one of them will be a cluster called P prime C. Given that said, we can take each one of those clusters and have only one matrix that I'm going to call here P prime main. Basically, what this P prime main does is I look to each one, as you remember, C is related to this C here. So the P prime C, so it's the number of different clusters that we can have. So I basically computing the average to each one of this points. So average here, here, here. And basically that's what I'm doing. And each row of the P prime mains would be that. So the average of the cluster 1, the cluster 2, and the cluster C. L prime is basically the number of lines that we're going to have in each P prime C. So as you can see, we can have, it's difficult to see how many, but have an idea. So we can have exactly the number of lines that we have here. So it'll be the number of rows in case of the P prime C. So each one of them, this is what the line is. So basically we can compute the average. So we're going to have those averages, as you can see, and we have M values. So we still have patterns with M observations. Given that said, we now have the following idea. Now that I compute the P mains, I can have these informations for each one of the clustered matrices. So the P prime C. And the idea is, given that we receive a new observation that acts new, we can have, it's important fact that the X new, a new observation coming from the field, has to be length equal N. So if you remember the data that I mentioned, it was weekly, so N would be N weeks in that context. It depends on the monetary system that you're looking at, but in our case would be weeks. So if we basically compute the association that come by a distance, that association that I mentioned, and with these association between each one of the rows from P mains are greater than a certain dpred, we're going to say that we have an outbreak, basically. But now, how can we compute this dpred? So dpred is defined for using each one of the P main rows. So basically, as a function of the number of patterns or the number of lines or rows that we have in each P prime C matrix. So what we have is this function, this is what the dpred is based. So it's a function of the number of patterns in each one of the clusters, and will be a d base, a certain base that we choose, plus 1 minus d base divided per the number of patterns to alpha. So basically, is the d stop base will be the baseline of the association matrix, the smallest that is allowed, and alpha is a constant that changes the shape of the function. Just to have an illustration to be more clear, we can see this is the plot of the number of patterns. So what we expect is, if we have many patterns in here, so given one group, for example, if we look just for this one, and let's say that this one has a lot of lines, lots of patterns. If they have lots of patterns, we can say, okay, dpred will be equal to the d base that we have. So in this case, d base equal 0.6. So as much as we have more patterns inside of one group, we expect that we'll tend to the d base that we choose earlier. So if we have patterns clustered that have not much patterns inside, not much rows inside, we expect to have the dpred tending to 1. So the alpha will just change the shape of this curve, and they all will be optimized during the training of the method. Well, given this said, we can have these parameters to be optimized. And the idea is, well, if I have a pattern that are occurring many times, if you observe that, we say, okay, I can use a d base as a predictor, as a baseline for saying that there is or there isn't an outbreak. If we're at low, not too much, I have to increase a little bit more. So I expect an association almost perfect or tending to perfect, depending on the value of alpha that was optimized. So that's the idea. So as a summary of what the method is, you will first have some variables relationship. So we have the first and the second step is, first we have to decide what is an outbreak or not. To do that, first you have to look at X star, for example. The X star will be the threshold that I mentioned that in the red line. And to do, again, it's related to the context that you have. So it depends on the species and the past that you're working with. In our case, with aphids, this is why we have 200, because we're considering all the species of aphids encountered in that particular example. So it will vary for species to species. And given that we have the outbreak, I select it here, and I select endpoints before the outbreak, and I form the P, that's called the D's matrix. After that, I select a certain D star cluster that will say to me, well, I want to cluster those patterns. I cluster them, take them in, and also as an output, before the output, I will have the function that I called to earlier, that is the D star pred. D star pred will use the D star base and alpha. And given that I have a naive observation, we will compute to each one of the rows of the P prime means, D's association. And if you have the association greater than the D pred, I can say, well, there is an outlarge. So with the outbreak is going to happen if otherwise we have no alert. So as you imagine, we have some parameters to be optimized. The first will be M. The second will be D cluster and alpha. So we have these 3 parameters to be optimized. So that's what we learn during the training of this method. And the D-base is going to be used to compute the ROC curve. So as I mentioned earlier, if we have an area under the ROC curve similar to 1 or tending to 1, the better the algorithm. So what we do is we're going to vary alpha, D cluster, and M, and have an objective function that computes the area below of the ROC curve. We do that by varying the value of D base between 0 to 1. And when we vary that, we will have the ROC curve being formed. So basically to each combination of alpha, D cluster, and M, we can have the leave one out cross validation happening. So what's the leave one out cross validation? We take the training set, but we know that there is an outbreak or not, and we use it to compute, try to predict those outbreaks. And using the leave one out cross validation, that basically we will just going back to the matrix in here, just for one example, I get rid of one line and use all the information on the other lines to predict this line here. And I repeat this process until the I. So this way I can compute values of true positive rate and false positive rate in order to form the area of the ROC curve. So what we use here is an algorithm called dual annealing, as we don't have a function to be to compute differentiation, to use gradient descent, for example. We don't have it here, unfortunately. So we need to have something that do not strictly need a function to be differentiated, as for example, multi-layer perceptron that use gradient descent to do so. We can't do that here. But this is why we decided to use simulating annealing or dual annealing. That's just a generalization of that. So basically, some of the values we have will be the alpha, M, and the area of the ROC curve, as you can see, going to 1. So of course, we have D clusters, D star clusters as well. And we vary those values, and using the dual annealing, we will select the one that has the higher value of the ROC curve, as you can see here. So basically, we're searching grid and using the dual annealing in order to compute and come up the best combination on a training set. So this is how the learning occurs in our algorithm. So basically, we're gonna go back to the data set. So the data set, as I mentioned here, we can do the 2 ideas. So split the time series in order to train. So when I mean train, I select the P prime mains. Also, I optimize alpha, I optimize M, and also I will be optimizing the value of the star cluster. And using those information optimized here based on the dual annealing, we use in the 70% for tests. So I go and try to predict if it is or it isn't an outbreak based on the lines. So as you can see, only using the P prime mains in order to do so based on this 30%. Also, we're going to vary for 50%, 50-50, train with 50 and test with 50. And finally, we train with 70 and with 30. Just one topic more, that the fact that here we have 2 time series. So one is decomposed, another is original. So we can have two values. So the next slide that I'm going to show you are the accuracies based on the original time dynamics and also in the decomposed dynamic. So we do that separately. All the process that I mentioned, we do separately, first for the original time series and the embedded. But the idea to select what we call the patterns are still based on the original. So just to mention, to clarify that, I walked through the observations, and instead of taking M observations from the original time series, I take M observations from the decomposed time series, and then I do all the process that I mentioned earlier. Well, given that said, we show the accuracy of the method that at the beginning with a 30% of training set have almost 77% of accuracy for the method with the time series with the original time series, or that there isn't decomposition in this case. And as you can see, with the one that has decomposition, have a little lower value of accuracy. But as I have 50%, I increase more the values of accuracy for the only using the time series original time series. But when I have 70%, the decomposed time series using the empirical mode decomposition got higher value of approximately 82% of accuracy. So it just shows you that at the moment to have an idea of the feasibility of the method. So we showed here the feasibility of methods of the proposed pattern-based prediction to predict population outbreaks based on time series data with a high accuracy, and we'll illustrate the method with the data that I just mentioned to you. So basically, at the moment, we're still working with other metrics to compute and compare the method with other available, but for that methodology, I would like to introduce showing this example and accuracy of the method. So in the end, I would like to thank Fundação de Amparo Pesquisa Estado de São Paulo, that was foundation for helping with financial support in 2019, and Agência USP de Inovação 2020, and now Science Foundation Ireland Centre for Research Training, Foundations Data Science for financial support that I luckily can keep working with the project and keep increasing the methods and producing more results. And I extend this thanks for my supervisor, Rafael Moral, Wesley Godoy, and the collaborators Olive Manson, Douglas Lau, and Eduardo Angel. So here is some of the references, and sorry for passing 1 minute from the 30-minute that I mentioned before. So thanks a minute for your attention. And again, thanks for the opportunity to be here. I'm very happy to talk to you. Thank you.
Thank you, Gabriel. Thank you so much. It was indeed very interesting project and very interesting methodology. I really enjoyed the talk.
Thank you.
But let's see if there's any question from the fellow and our attendees here. So is there any question? You can just raise your hand or share your audio and ask question. Or if you are comfortable, you can type in the chat box. Okay, no, it seems no question. I was listening and I had a few questions, but then they were answered by your presentation over different slides. So, but one main question I have is, so I really like the application of your methodology, especially in this domain, which I think is quite helpful. But do you see any other application in any other domain that this methodology of prediction for the time series data can be used for?
Oh, fantastic. Thanks, Amina, for the question. Yeah, what the method is basically what we look is binary problem when we have patterns from the certain event. So the event particularly would be an outbreak in our case, but I think we can still extend to other applications here in biology as well when we have binary problem as well. But the main application of that, as I mentioned, with this problem, with where it came from, was in 2011. So as I mentioned, the Richter map was the author that introduced that idea in 2011. He used the Richter map just to show the patterns, to say, oh, look, we have here from the Richter map that basically that was the main output of his work, that we can still have some patterns here. And he didn't mention anything about prediction. So the inspiration of the project comes from this paper of 2011. I think since the beginning of the project, I've been thinking about that patterns from outbreak, but thinking here, I think we could extend to other applications, just need to carefully look what have in the literature, because the method is looking to the numbers itself, and we're optimizing them based on the patterns. But I think other applications could be suitable, just as a matter of looking for it. And thanks a million, I'm going to have a look for this.
Oh, no, yeah, that's fine. We have one question here from Paddy. That was my question too. So the question is, in model training, did you consider using some kind of rolling cross-validation, given that your data is a time series?
Cross-validation, yeah, we look, so basic, I can go back here. You mean the cross-validation on the training set, isn't it? So is it? Oh yeah. So this part we'll look to in the training set, what we're doing to compute the accuracy and to compute the area below the rock curve is basically leave one out cross-validation. So we do in cross-validation here in order to do a generalization. So we select the part, as I mentioned earlier, we do in each one of the part of the observations that we have, we will look in each one, we don't consider it for the part of the matrix, compute the active value, if you can predict or not, move to the next, so on and so forth. So that's would be the leave one out cross-validation working on the training set. Was that the question? Did I answer that? Hopefully.
I'm not sure. Maybe Pat, you can help us. But I think, yeah, that's covered. My main question, my idea was actually, I don't think that may be applied here, but I was thinking if you could use the model to retrospectively predict the data. For example, for most of k-fold cross-validation, you split data into k-fold and then use one of the fold as test and the rest as training. But I don't think that can be applied here because you cannot use future data to predict the past. Can you? I don't think so.
Oh, you think, oh, perfect. You mean like instead of using 70% or 30%, I can use K sections of this.
Yeah,
that.
Yeah. But for example, if you use the last 9, if you use 10-fold cross-validation, you use the last 9 section as training to predict the first section or first fold, which I don't think that may make sense.
The idea was we at the beginning when we thought in this way of splitting was mainly because of the order of the observations and try to maintain the structure of the time series itself. But yeah, I'm trying to remember if I've seen that before in any paper, but I don't think so. But thanks a million for that. Really, but the cross validation we do here in this part, but I meant the cross validation for this matrix here. Oh, for this matrix, that it will make more sense in this case because there's not the structure for them. So in this part here, that will be the events itself and the patterns itself. So here we can do the live on our cross validation. We will still be respecting the time series, the thinking.
No, that makes sense. That totally makes sense to me, at least. Yeah.
Good.
I had also one question from this matrix here. So what happens if 2 events are so close together and the 2 rows be correlated with many observations the same? Does it affect your result?
Oh, you mean correlated? These 2?
For example, yeah, let's say the first 2 outbreaks happening in a very short time. So therefore, you expect the past M measurements somehow be very similar, at least, let's say 4 or 5 of them, depending on whatever the value of M is. Does it affect how your model, you think?
Well, it would be one of, if we, in a short answer would be yes, because if we have, for example, this, we tried before, as I mentioned earlier, we're doing this association, right? Before we tried to use different associations, for example, correlation between them, right? Instead of using here, the C would be correlation to have this pattern. So as I've seen before during experiments, sometimes when we have 2 patterns very related together, you could affect a bit the accuracy of the method. So it will with something, it can be, but it's not that clear. I can't say to you as a linear or it's not linear or what is happening between these. If we have, for example, the accuracy between them is the correlation between them are much higher. it will affect the end of the accuracy of method in this degree. I can't say that, but I noticed through simulation, we did more, I couldn't have the time here to show it to you, but we did some other simulation analysis, and depending on the structure of that matrix, it definitely affects. I don't know how in detail, but I could say in short, yes.
Okay, that's good. No, that was fantastic. So I really enjoyed, and I'm sure that our audience, they also enjoyed the presentation and the talk. So thank you so much, Gabriel. So let's see if there's any final question from people here. Otherwise, we are off to go. So any final question? Right. No, there's no question. Thank you so much, everyone, especially Gabriel, for a very nice presentation. Hope to see you all in person soon. Thank you, guys. Thank you all.
Sure.
Bye.
Thank you. Thanks a million. Bye.